how can we know if your friend's plant is doing more photosynthesis than your plant? So in essence, we know that three factors determine how good a plant does photosynthesis. The more light, the more photosynthesis. The more carbon dioxide, the more photosynthesis. And the optimal temperature means optimal photosynthesis. But how can we measure photosynthesis? Like, how do you do that? You can't measure photosynthesis with a ruler or with a thermometer. So how do you measure it? Okay, that's what we're going to be discussing right now. So there are three ways that we can measure photosynthesis, and those include biomass, bubbles, and pH. Okay, let's go on to the first and easiest one, biomass. We're going to be using this little image here to explain this whole process, okay? So we have Captain Jack Sparrow stranded on an island, a nice beautiful island, and we're going to zoom into the water and look underneath, okay? And this is what we see. We see a plant with some rocks on the sand and a little squid or octopus. I'm not sure what this one is. And we think biomass is the same word as human mass, basically. Human mass means how heavy a human is, right? The more we eat, the heavier we get. Now, biomass is a word to describe how heavy a plant is. So the more a plant eats, the heavier it gets, right? And we know through photosynthesis, a plant makes glucose, right? So here I have a little image where I explained photosynthesis in a previous video. So you can check this one out if you don't understand photosynthesis yet, because that might be important. But anyways, we know photosynthesis creates glucose. Glucose is food, okay? And so the more food the plant makes for itself, the more it's going to use. So the more it's going to grow and have bigger leaves and become heavier. So therefore, we know that biomass is important because the heavier the plant gets, the more its biomass is, it means that it had more photosynthesis. So for example, if your friend's plant and your plant started from baby, and his plant is the same species as your plant, yet his plant weighs 500 grams and yours weighs 1 gram, then obviously his did more photosynthesis, right? Because his biomass increased more than your plant. So, but the problem is biomass takes time, right? We have to wait for the plant to grow and then compare and measure and all these things, right? Now, let's go to bubbles. This is where it gets a bit more complicated. So here we have this plant under the water. And the reason why I put it under the water is because you'll see why. Because the outside plants, this doesn't work that good with. So we're talking more about aquatic plants now. It is the same thing for terrestrial plants, but you'll see why it doesn't work so well, this experiment. So we know that through photosynthesis, right? So here's photosynthesis. We absorb sunlight into the chloroplast, specifically this little structure here called the thylakoid. And this sunlight will split water. All this energy from the sunlight will break water apart into its constituents, into hydrogen, oxygen, electrons, and ATP, right? Through a process called photolysis. Now, basically, in the process of creating these constituents, we see one of them is oxygen, right? Oxygen is the air we breathe, okay? We know oxygen is in the air. Now, oxygen underwater is a bubble, right? So if this plant is doing photosynthesis, it is going to create a lot of oxygen. Only oxygen forms bubbles. Okay, so let me show this. So basically, it's going to create a lot of oxygen, okay, because it's releasing it. But in a terrestrial plant like this one, we can't see this oxygen being created because it's in the air. We can't see it. We can't see oxygen. But underwater, oxygen forms little bubbles. Because it's like when you blow out the air from your mouth, you can create bubbles underwater, right? That's the same thing with a plant. If it's creating oxygen like this, so this blue bubble here represents oxygen, then we can just see how many bubbles there are. So for example, you shove your plant maybe underwater and your friend's plant underwater. Hopefully they're aquatic, otherwise they won't survive. But if they are, you can just count the bubbles. If your plant is making more bubbles or more oxygen than your friend's, then it's doing more photosynthesis. It's simply that simple, okay? So that's one part of bubbles. So that's half of what I'm talking about with bubbles. There's one more thing with bubbles that's important, but it links to pH, okay? So bubbles, just think about bubbles as oxygen. The more oxygen is created, the more bubbles you can count, the more photosynthesis the plant did. Now, let's move on to pH then. So we know, let me delete this oxygen. I'm going to delete the oxygen or just move it back down here. Oh yeah, I forgot to use this, but this was the image that I was going to use to show biomass, right? The more photosynthesis a plant does, the bigger it's going to grow. So here the plant grew a lot, okay, because it did more photosynthesis. It can increase in size. Now, pH. So pH is a bit tricky to understand. We know, right, through photosynthesis, we use carbon dioxide, right? Humans can exhale carbon dioxide, but plants can use it. And plants, interestingly, exhale oxygen, as I just mentioned, but humans can use it. So we work together. Now, we exhale carbon dioxide because the plant is going to take it up into its little chloroplasts to combine it with all of these materials produced during photolysis. And when it's combined together, it will form glucose, right? So carbon dioxide is consistently taken up from the environment by the leaf cells, okay? By these leaf cells. So if we were able to tell how much carbon dioxide is being taken from the air, we would know if photosynthesis is happening a lot or not, okay? So let me show you. So we're going to take these red bubbles. These represent carbon dioxide, okay? The blue bubbles were oxygen, now it's carbon dioxide. So this plant, if it's doing photosynthesis, it will take up these little red bubbles or carbon dioxide, and it will use it to do photosynthesis. Now, carbon dioxide is an acidic compound. What do I mean by this? Here is the pH scale, okay? Acidic. Acidic is anything that has a low pH, okay? These kind of things will burn you if they touch you. But carbon dioxide is a really weak acid. It's really weakly acidic, so it's like around here. It's not that acidic, but it's still acidic. So with a pH meter, you can measure the water. So if there's a lot of carbon dioxide in this water, you can stick a pH meter in here and see what the pH is. A lot of carbon dioxide would mean a low pH. So maybe you'll be at like a pH of 3. If this plant is taking up this carbon dioxide the whole time, it means the carbon dioxide is getting reduced. So it's removing all the carbon dioxide from the environment. If it's removing all the carbon dioxide from the water by doing photosynthesis, then what will happen to the pH? There will be less carbon dioxide, and we know carbon dioxide makes it acidic. If there's less carbon dioxide, then the pH will shift to the right. So it will move from like pH 3 to maybe pH 4 or 5. So the pH will increase during photosynthesis. So again, you put your plant into the water, and you put your friend's plant into the water, you measure the pH in the beginning, and then you measure the pH 10 minutes later. Whoever's pH increased the most means their plant used the most carbon dioxide, and so your plant would have done the most photosynthesis, okay? So that's it. Those are the 3 very important factors or ways that you can measure photosynthesis. Biomass, how heavy the plant is, bubbles, how much oxygen is produced, and pH, how much carbon dioxide is used up, because pH is highly affected by carbon dioxide.